Assalamualaikum and good day everyone. Today we're going on with gene regulations, especially in prokaryote. But before that, probably we'll see some of the similarities and differences between prokaryote and eukaryote so that you can, differentiate them better. But before that, again, let's see the objectives of this very simple topic. So it's basically just to describe the role of protein and DNA interactions in regulating transcription initiation in prokaryote. We're also going to more or less hopefully distinguish positive regulation from negative regulation. So this is where next week we will go into operon. The third one is to identify similarities and differences in gene regulation in prokaryote and eukaryote, including the mechanisms of gene co-regulation, presence of chromatin in eukaryote, and post-transcriptional regulation in eukaryote. That would be after week 7. And the last objective is to use a gene regulatory system model, such as the lag operon, to predict the effects of mutations in various components. Alright, so basically, from last week, and if you have seen the video on the basically overview, there are a few levels of regulation of gene expression. There are actually 7, but over here I'm highlighting 3. Transcriptional control, which is where gene is being transcribed into mRNA. Translational control, whereby mRNA is being translated into protein. And post-translational control, including active or inactive but stable or degraded protein. Now, a rough basically outlook on the regulation of gene expression regardless of whether it's in eukaryote or prokaryote. There are always positive regulation and there are also negative regulation. Positive meaning that turning on gene expression, and of course negative would be the other way around or the opposite, turning off the gene expression. So apart from the positive and negative regulation, we also have co-regulation. So the name co would suggest that you could turn on multiple genes on or off together. So you could control more than one at the same time. And apart from all these 3, some genes are actually not subjected to regulation at all. They are called constitutive genes, which are always transcribed. Now, let's have a look at similarities between prokaryote and eukaryote. So among the similarities between both of them have promoters and regulatory elements. I think probably in the second video we will see the outline, or basically the linearized version of a gene whereby you can see the promoter and regulatory elements. But if you could basically refer to a book or just Google on the outline of the promoters and regulatory elements. For the promoters, usually it is available at the DNA sites, and usually you do need this promoter to start off a regulation for positive regulation. And this is where it's going to get a bit confused in like operon or the trip operon or the error operon, as you will see. So apart from promoters, there are also RNA polymerase, and the function is to bind to the promoter for it to initiate transcription, because if promoter is there but RNA polymerase is not there, initiation of transcription will not occur. You cannot get your gene to be transcribed. Apart from promoter and RNA polymerase to start off the transcription, the gene also contains binding sites for transcription factors. Now, in prokaryote, probably we're not going to go into details on the transcription factors because they are general transcription factors. But in eukaryote, you will see that there are at least 6 of them that have to come subsequently in an order, whereby if they are not in order or if one of them is missing, the gene will not be transcribed. So secondly, let's have a look at the regulatory elements. So what are they? They are basically DNA sequence which is recognized by the transcription factors and will bind to it in order to recruit or repel RNA polymerase. So this is always true in prokaryote because there are certain sequences that will be recognized by the RNA for the transcription factors. So it could be used for either a positive or negative regulation, and both positive and negative at the same time. So now I have more or less a question here. As you can see, okay, there are question marks here. So some genes, if you remember in a few slides before this, okay, are not subjected to regulation. They are constitutively expressed, meaning they are always transcribed. So anyone could just think of, okay, in your mind first, okay, or just jot it down, okay. Probably when we meet next, we will discuss it a bit. What sort of genes would you imagine a cell would always need to have on, regardless of the environment or situation? Think about that. Either in prokaryote or eukaryote, or you yourself, a human. Think about it. So while you think about it, let's focus on prokaryote, since this whole 6 weeks plus last week is basically just for prokaryote. So how about bacterial constitutive genes? What are they? What are some of them? Why do we need them to be constitutively turned on or expressed? So I have some examples. This is from a journal from 2015, as you can see it. Okay, so from Van Leeuwenhoek 2015 et al. So the most commonly tested and validated bacterial reference genes identified by this study. So they are also called as housekeeping gene. Ever heard? Yes? No? For those of you who have done your undergrad project on 16S RNA, probably you would have heard of this, or do some molecular work where you do some PCR, you may have heard of this. We use this housekeeping gene a lot, basically just kind of like to see again the quality of DNA or RNA. So these are some of the most common constitutively expressed genes in bacteria. 16S rRNA, okay, gyre A, okay, which is DNA gyrase A, and so forth. Okay, these are some of them. There are a whole lot more. These are the most famous, simply said, okay, and have been studied, tested, and also validated, as you can see. So there are a whole lot more. And can you imagine, in one bacteria, usually they have more than one gene which is constitutively expressed, regardless of the environment, regardless of the condition of the bacteria itself. Why? Usually this is to survive and for their survival. Now, whatever that I've asked you in the previous slides, okay, so think about it, okay, when we meet next, like I said, okay, we will discuss, and you may also want to discuss or highlight something that is at the back of your mind and whatnot. But before that, okay, before we come to an end of the first video, let's have a look at the differences between prokaryote and eukaryote. So in the previous slides, we have seen the similarities. So now we're going to have a look at the differences. So what are the differences? Stark differences, of course, in the mechanisms of co-regulation. So what is co-regulation? So co means that basically you can turn on or off the genes at the same time. And of course, this is in response to the similar or the same stimulus. In prokaryote, genes are co-regulated via operons, because we know operon is mostly simply said the main mechanism to control gene expression. And in prokaryote, single promoter can be used to transcribe more than 2 genes into one mRNA called polycistronic mRNA. Oops, the arrow is not moving. But okay, the same mRNA may be translated into a few proteins. So we only have polycistronic mRNA only in prokaryote, not in eukaryote. Now, in eukaryote, there are no operons, except for C. elegans and related nematodes. And I think starting from 2019, they actually have found in a few fungus as well. So DNAs in eukaryotes are organized around histones, which is referred to as chromatin. I'm not going to go into details, okay, on the histone or chromatin. You need to read up about it, okay, basically how histone, okay, that basically binds or forms the chromatin, okay, is actually crucial, okay, to regulate the mechanism. And in eukaryote, the co-regulated genes, even at different chromosomes, could basically be turned on or off using the same regulatory element sequence. So can you imagine, okay, these genes, again, they are situated at different chromosomes. So for example, as human, we have 23 pairs of chromosomes. So even if they are, for example, at chromosome 1 or at chromosome 19, so they could actually be regulated using the same regulatory element sequence. How cool is that? So I basically have summarized this for you guys. So overall differences and similarities in gene regulation between prokaryote and eukaryote. So if you can see it here, if you were to print it out in black and white, you may not see it, but if you were to view this on your computer over the internet, you will see that the differences and similarities are being written in different font color. So I have blue, I have green, and I have basically black. This is actually also black. I'm not sure why it's kind of look like gray. Okay, so now we have couple transcription and translation. We have 5' cap and 3' poly A tail. We also have AUG as the translation initiation codon. We have regulation of gene expression by proteins binding to DNA regulatory elements. We also have alternative mRNA splicing, and we have regulation of gene expression through chromatin accessibility. So, which is the similarities, which is the differences, and if they are differences, again, whether they are basically just only in prokaryote or in eukaryote. So hang on to this slide, think about it. Okay, we will have a discussion when we meet next. So, well, this is the end of the first video. Move on to the next video.